Hello, everyone, and welcome to today's webinar, Deep Immunophenotyping of Cancer Microenvironments by Imaging Mass Cytometry. I am Tracy Aston Martin of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is sponsored by Fluorescent. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Ask a Question box located on the far left of your screen. I now present today's speaker, Dr. Noel de Miranda, a Principal Investigator in the Department of Pathology at the Leiden University Medical Center and a project leader for the Immunogenomics group. His group aims to develop innovative therapies that are focused on stimulating a patient's immune system to recognize and eliminate cancer cells. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Noel, you may now begin your presentation.
Many thanks for the introduction. I will indeed show a bit of our work where we've been applying imaging mass cytometry to characterize the microenvironment of cancers, with a particular focus on interactions between immune cells and tumor cells. I will start by acknowledging the people that are actually doing the work. Marika Eiselstein is the PhD student that set up the Imaging Mass Cytometry panel for FFPE tissues at the LUNC. And Antonio Somarakis has developed a number of image analysis tools that will be discussed in this presentation. We also work in close collaboration with the groups of Fritz Koning, who has really been instrumental in bringing this technology to the LUNC, and Biography Lilifeld, who has the Division of Imaging Processing at the LUNC. So why focus on cancer microenvironments, and in particular in the immune cell component of cancer microenvironments? It is now well accepted that the quantitative and qualitative composition of the immune cell compartment in the tumor is a major determinant of clinical behavior, with the presence of different immune cells being associated with either improved or worse clinical outcomes, depending on which immune cell subset you're looking at. And there's really been a renewed interest in cancer immunology in the last decade, as a result of the successful development of therapeutic antibodies targeting co-inhibitory receptor in T cells, like the ones mediated by PD-1 and CTLA-4 receptors. These therapies have been particularly successful in tumors with a strong immunogenic profile, which is connected to the accumulation of large numbers of somatic mutations throughout their genome. The elevated number of mutations then translates to an increased likelihood that T cells will be able to identify and eliminate tumor cells. Accordingly, what you also see is that tumors with more mutations are generally more densely infiltrated by T cells, and in particular by CD8 positive T cells. The determinants of response, however, are not limited to mutation burden and infiltration by T cells, as it can be deduced by the fact that only a minority of patients with theoretically immunogenic tumors are currently responding to checkpoint blockade therapies. In fact, as depicted in the cancer immunogram put forward by some of our colleagues at NCI, together with Anthony Rivas, another model that in the meantime has been proposed, there's really a myriad of mechanisms at play that influence immune responses to immunotherapy and which need to be considered, that include the presence and functional state of other immune cell subsets than just T cells or cytotoxic T cells. So it has been very exciting to see throughout the years how technology has evolved and made it possible to look deeper into tumors and to multiple features simultaneously. By both methods, in particular with bulk RNA sequencing, we can assess transcriptional programs of cancer, and groups have developed tools to inform on immune cell composition of tumors based on these data. However, such analyses are restricted to averages or approximations of immune cell really allow for the investigation of complex phenotypes, and certainly not at single cell level. More recently, single cell sequencing and other single cell technologies have really been a major breakthrough in the sense that they provided us with the ability to characterize in detail the phenotypes, but also genetic features of single cell. However, one must not forget that such technique does not include the spatial contexture of cells, and there are biases associated to our ability to retrieve certain immune cell subsets from tissues. Particularly when we're talking about solid tumors, it can be quite challenging to retrieve all subsets that are contained within the tumor microenvironment. So to address these insufficiencies, it is fundamental that we can make use of multiplex imaging technologies that not only inform on the phenotype of the cell, but also their location within the tissue and the cellular context they're inserted in. Several groups like the ones, for instance, Jérôme Gallon with Immunoscore have now demonstrated that different tumors can carry the same amount of T-cells, for instance, but their differential localization can have profound clinical implications which would not be picked up by a method that ignores spatial context. We are therefore very lucky that we can make use of an imaging mass cytometry instrument at LMC, where the parallel detection of several antigens can be done simultaneously by making use of heavy metal labeled antibodies. So in the past years, we've been working very hard to set up a panel of around 40 markers. And the description of this process was published at the end of last year. We went ahead with labeling each and every clone that was previously tested by us by standard immunohistochemistry. And this is because we wanted to have the flexibility to combine specific antibody with particular metal tags. And during this process, we realized a few important things. First, that a mixture of 40 antibodies did not perform very well in one go, and that antibodies also perform best in different conditions, different types of antibodies. To address these two issues, we made use of two sequential incubation steps, one overnight at 4 degrees and the other at room temperature, and we distributed the antibodies by these two conditions according to their performance, and thereby also splitting the total amount of antibodies per two incubations. So our current panel includes a number of immune cell lineage markers, so it has been updated since the publication of our paper. It includes a number of immune cell lineage markers, differentiation and activation markers, immunomodulatory molecules, including therapeutic targets, some signaling molecules, and finally, and also very importantly, some structural markers that allow us to map the tissue for the different components as well to distinguish epithelial from stromal layers. We make use of a number of in-house developed tools, as well as tools that are publicly available for the processing of the data. So we start with inspecting the quality of the antibody signal with the MCD viewer, and if that looks good, we develop cell masks and perform background subtraction by making use of cell profiler and elastic. We then transform this imaging data into a per cell matrix that is used as an input for CyTOF, where with TSNA-based dimensionality reduction, we identify cell clusters that can correspond to cell subsets. The biological validation of such subsets and further analysis are then conducted with ImaCyte. Just to give you an example, with ImaCyte, we can inspect the presence of a particular cell cluster in the masks that we apply to the images and evaluate whether that cluster has indeed the morphology and distribution that was expected. So a tumor cell should look like a tumor cell, a myeloid cell like a myeloid cell, and a lymphoid cell like a lymphocyte. A lot of our work focuses on colorectal cancer, and you can divide colorectal cancers in 4 major molecular subsets according to their transcriptional profiles, where you have a group that shows high immunogenicity and consequent infiltration by effector immune cells, the CMS1 type, the CMS2 and 3, which are subtypes characterized by very poor immune cell infiltration and driven by Wnt and MYC activation and metabolic dysregulation, respectively, and a 4th subtype which does demonstrate some type of inflammatory process going on, but the microenvironment is largely dominated by ATG beta stromal signature. we wanted to know whether we could see such transcriptional profiles reflected on the analysis that we conducted with imaging mass cytometry. We processed some samples by this methodology, and through the analysis approach that I explained before, we defined the following clusters where I confess we adopted a rather conservative approach to avoid defining too many subsets. So we identified, for instance, 21 tumor cell clusters, 13 different lymphocyte cell clusters, 14 myeloid clusters, 8 stromal subsets, and 7 types of cells which we cannot really give an easy biological meaning to those. So despite being rather conservative, we thought this number of subsets already gave a nice structure and complexity to the data set to perform subsequent analysis. So some of the findings were not very surprising, namely that T cell infiltration, particularly by activated cytotoxic T cells, is really a hallmark of the CMS1 type. But perhaps a bit more surprising is the fact that such immune reaction is accompanied by a very profound infiltration by granulocytic immune cells. And we still really have to understand what is the meaning of their presence and how they intervene in the context of checkpoint blockade therapy, for instance. And one of the coolest things of this technology, in my opinion, is our ability to distinguish tumor-associated myeloid subsets, namely different flavors of macrophages, monocytes, dendritic cells, which I think is quite challenging to do with other technologies, namely single-cell technologies. And it's really nice to see in the images the complexity, or we know that these subsets are really complex, and it's very nice to see in the images, indeed, all these different shades of myeloid cells, which have not always been captured with previous approaches. Finally, it is also extremely interesting to study how the accumulation of immune cells associates with features of tumor or stromal cells, for instance, the relation between immune cell infiltration and the presence of TGF-beta related molecules in the microenvironment. An additional bonus of the multiplex technologies is that it also allows you to discover things like chance and stuff that you were not really able to immediately explain based on current knowledge, like it is the case when we find subsets through the combination of markers that were not anticipated to go together. And here, of course, you run the chance of being looking at artifacts, so one really needs to make sure we validate whether these subsets are really meaningful in distinguishing a true immune cell population. But once we see that these are typical particular subtypes of colorectal cancer, we think there might be an interesting biological meaning to that. Finally, the imaging also allows us to investigate in depth what is the relation between all subsets that have been identified, and whether differences in the spatial arrangements of cell subsets also play a role in pathogenesis. And this is a glimpse of what we've been doing in addition to numerous projects that are currently ongoing in combination with the clinics to try to extract information that is valuable in different tumor types, and particularly in the context of checkpoint blockade treatments of cancer patients that are undergoing immunotherapy. In the coming years, we will probably be working very hard on the integration of data sets derived from different multiplex platforms, and we think that IMC can really play an important role in, for instance, supporting the mapping back of single-cell data, or the mapping of single-cell data back to tissues. And I'll finish my presentation here the same way I started, by acknowledging the people that are actually doing the work, and I'm more than happy to answer questions that might come my way. Thank you.
Thank you, Noel, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. So let's get started. Let's see what we have for questions. We have quite a few questions coming in. We'll start with our first question. Have you been using IMC for other purposes than immune oncology?
Yeah, so the lab has been employing imaging mass cytometry in other projects as well. Basically, any setting that requires the use of multiple markers to assess a phenotype of a cell. So for instance, we also have a project that is focusing on signaling. And there what we have built is a panel that is mostly constituted by phospho-specific antibodies to study signaling in tumors.
Our next question. How do you see the integration of single-cell sequencing with IMC going forward?
Yeah, so as discussed in the presentation, there are some advantages to using single-cell technologies and others to using imaging mass cytometry. But in the lab, we also see both as highly complementary. So what we've been trying to do is to project the single-cell sequencing data on gene expression back to the images. What we do is you use common markers between the single-cell data and the imaging data, and you determine the probability that a certain cell from single-cell sequencing belongs to a certain part of the tissue. And this is easier to be done if there are some common markers, for instance, in the setting of CyTOF, where you could use the same antibody that you use for IMC as that you use for CyTOF. And these are attempts to try to increase the amount of information we can get from a cell by joining these gene expression profiles or with their spatial localization.
Thank you, Noel. Our next question. Do you develop the analysis pipeline yourself, a custom solution, or use something that is commercially available?
Yeah, so for the cell masks, for creating the cell masks and for determining what's real signal and what's background signal, we use tools that are available, like Cell Profiler or Elastic. But we are lucky enough that we have a division of image processing at the LEMC, and that's a research group on itself. So we're very lucky to be able to rely on those guys to develop solutions for the analysis of data. But there are also tools which are available off the shelf for the processing of IMC data, like the ones developed by the Bodemiller lab, like HistoCat, for instance.
Okay, thank you. Is IMC applicable to other tissue types or just for FFPE?
It really shouldn't matter. So we also use imaging mass cytometry panels on SNAP frozen tissue. What one has to realize is that the panel of antibodies will not be the same for FFPE and for frozen tissue. And that has to do with the availability of the antigens. So for instance, if you're used to use applications like flow cytometry, if you want to use the same clones, those will be more likely to work on frozen tissue. While for FFPE, well, you probably have to select a number of specific clones that will only work on FFPE and not on frozen tissue.
Okay, wonderful. Well, it looks like that's all the time we have today. Thank you again, Dr. Noel de Miranda, for your time today and your important research. Before we go, I'd like to thank our audience for joining us today and for their interesting questions. Oh, we just got one more question that just came in, so I'm going to go off the rails and ask this question. This is an important question. Are the metal conjugation kits easy to work with? Did I pronounce that correctly? Are the metal conjunction kits? Okay, sorry.
Conjugation, yeah.
Conjugation,
so,
okay, thank you.
we do all the labeling ourselves. So we had to rely heavily on the conjugation kits. I think it's quite straightforward to use. I mean, as everything in the lab, it requires a bit of accuracy, and it takes a bit of time, but it's quite straightforward. You should be able to easily do it yourself. It's just following a protocol. And of course, you need to get the antibodies on a special formulation because they cannot have stuff like BSA or other proteins. Otherwise, we'll be labeling those rather than the antibody itself.
Okay, great. Again, thank you again, Dr. Miranda, for your time today and for your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions that we did not have time for today and those that are submitted during the on-demand period will be addressed by our speaker via the contact information you provide at the time of registration. This webcast can be viewed on demand through July 2021. Until next time, be safe, everybody. Have a great day and goodbye.